Lipids are our fourth class of organic macromolecules. And I left lipids for last because this is the one class of macromolecules that's different in a couple of ways from the other three classes. One, lipids are not analogous to beads on a string. They're kind of an odd collection of structures. But what they all do have in common is they are primarily hydrophobic. So carbohydrates, proteins, nucleic acids, these are all beads on a string. So we had examples of polymers built up by monomers held together by specific types of bonds. And they're all hydrophilic, so lots of polar covalent bonds found in all these molecules. Lipids are hydrophobic, meaning that they are primarily made up of carbon and hydrogen. And so these are the fats, oils, and waxes that we're familiar with. These fats, oils, and waxes have a variety of functions. The primary function that we're going to talk about is energy storage. But we're also going to focus some on the structural lipids, those that give the cell structure. And then we're also going to talk a little bit about molecules that are considered lipids that are involved in signaling. So hormones. I talked a little about peptide hormones when we talked about proteins, but there are also lipid hormones. So like I said, these are the only class of organic molecules that are considered to be primarily nonpolar and hydrophobic. And we're not going to see polymers here made up of individual monomers. Now, there isn't any molecule that works in the cell that's completely nonpolar, and that's the case for any of these lipids. But the first type of lipid that I want to talk about are the triglycerides. And to do that, we first have to talk about fatty acids. Fatty acids are long hydrocarbon chains. The length of carbon is most commonly 14, 16, or 18. So we always see these carbon chains in groups of 2. And the long chain of carbon and hydrogen is terminated with a carboxyl group. This is a fatty acid. Now, in 2 of the types of molecules, triglycerides and phospholipids, we're going to see these fatty acids attached to a molecule of glycerol. This is a molecule, glycerol. It has 3 alcohol groups and hydroxyl groups, so it's an alcohol. And the rest of it is hydrophobic. So when we join fatty acids to glycerol, we're going to definitely have a dehydration reaction, but this isn't a true example of monomers strung together to make a polymer. So the first type are the triglycerides, and triglycerides are made up of 3 fatty acids attached to glycerol. Now let me talk about the types of fatty acids that you're going to find. Fatty acids can be either saturated or unsaturated. Saturated fatty acids are those where we have single bonds in between all of the carbons. Wherever we don't have a bond with carbon, carbon is bound to hydrogen. Unsaturated fatty acids are those where there's a double bond in at least one place in the chain of carbons. So in unsaturated fatty acids, the structure is a little bit different. There's a kink wherever you see the double bond between carbons. And notice that the carbons aren't completely surrounded by hydrogen if they're involved in a double bond, because you could only make 4 covalent bonds. So we say that the carbons aren't saturated, and that's why it's called unsaturated. You can have saturated fatty acids with no double bonds, monounsaturated with 1 double bond between carbons, or polyunsaturated where there's more than 1. And if you take a look at what like a whole row of saturated fatty acids might look like, you can imagine it almost being like a perfectly flat stacked sheet of fatty acids. But if we were to create a row of different types of fatty acids, a mix of both saturated and unsaturated, notice how it's not a very nice flat row of fatty acids. Everything that's boxed here is a kink produced by double bond between carbons. This is going to be important when we look at the types of fatty acids that are found in triglycerides. And here I have a picture of a triglyceride. 3 fatty acids, that's the tri part, attached to a molecule of glycerol. At each of those hydroxyl spots, a fatty acid was attached. Notice this has 2 saturated fatty acids and 1 unsaturated fatty acid. And the combination can be any in a triglyceride. Now, in butter, which is animal fat, we tend to find the triglycerides made up primarily of saturated fat. In plant oils, like olive oil, the fatty acids that are found attached to the glycerol molecules are more often unsaturated. What this does is it allows the lipids in plant oils to be more fluid at room temperature or at any temperature. This is going to be more fluid than this type. That's because these fatty acids, these hydrophobic chains, pack together really tightly, and they are attracted to each other because of hydrophobic forces. Here we've got kinks, and wherever there's a kink, you're not going to have a nice flat stack. So there's space in between this and another fatty acid, which is just going to increase the fluidity in general. Triglycerides are the form of energy storage. Animals store up long-term energy. We store up about a month's worth on average in fat. Plants also store up some energy as lipids. We store up more energy as lipids than we do as carbohydrates because lipids are lighter. They tend to exclude water, and water would make it more heavy. They also have lower molecular weight and so tend to carry more energy in those carbon and hydrogen bonds for less weight than do carbohydrates. And we need to carry around our energy. And so if we were to carry around a month's worth of energy as carbohydrates, we'd probably be on average 50 pounds heavier, which with our body structures is too much. Now plants, they don't need to travel with their energy. So most of the energy stores in plants is in the form of starch, not so much in lipids because they don't have to carry it around, so they don't care if it's light or not. And they tend to store it up in these unsaturated fatty acids, which are a little bit easier to break down and also not quite as much energy storage as these saturated fatty acids. If you take a look at a table of all the different types of fatty acids, notice that the animal fats tend to have, on average, more saturated than unsaturated fatty acids. And you can take this as a percent because it's grams out of 100 grams, so 40.8% saturated fat. And butter, 54% saturated fat versus the unsaturated. Vegetable oils, on the other hand, with the exception of coconut and palm oil, vegetable oils tend to be more unsaturated fats. Now, this is important because saturated fat increases your risk of heart disease. A relative risk of 1 means that there is no difference in the risk of coronary heart disease, is what we're looking at here, when compared to an equal amount of carbohydrates that are taken in. If the risk is greater than 1, then there is an increased risk. And so saturated fat, if you were to like let's say originally you start with half fat, half carbohydrates for your energy intake, and your risk goes up every 5% of your energy that's taken in as lipids. And notice that in trans fats, for trans fats, it's even worse. So what we want to do, and I'll talk about trans fat in a minute, what we want to do is keep our intake of fat in mono unsaturated and saturated, polyunsaturated fat. That's because this stuff is just as likely to pile up and stack up in your blood vessels as it is at room temperature. I mean, it's a little bit warmer in there, so it's a little more fluid, but this is what clogs arteries and causes heart disease. And unsaturated fat will not do that, and actually in some cases lowers cholesterol, which is another lipid that we're going to talk about. Trans fat is just when it's manufactured fat. There are some examples of naturally occurring trans fat, but trans fat is basically comes about by hydrogenation of vegetable oils. So oleic acid is a vegetable oil. It's a fatty acid, I should say, and it's found in vegetable oils. And it has a single double bond between 2 carbon atoms. When hydrogen is added in, that is then becoming hydrogenated, and it's a way to make vegetable oils a little more solid. So when you use buttery type spread that sounds good because it's made out of vegetable oils, it's really hydrogenated vegetable oil, which acts just like saturated fat in your vessels and in your heart. So this results in something that's like a saturated fat. And also, you remember when we looked at double bonds between carbons, we saw like two hydrogens on one side of the plane, and then there was this empty spot representing the unsaturated region of these carbons. Hydrogenation can do something else besides just making hydrogenated fat. It can make trans fats, and trans fats have this weird orientation. Here are the cis fats. The trans have these weird orientation of the hydrogen. These have the same numbers of oxygen, carbon, and hydrogen as our unsaturated fatty acids, but because they've been manipulated, these are now in trans configuration, and they also act as saturated and actually have been shown to be even worse than saturated fats. So hydrogenation is changing vegetable oils to a state that's not going to do you any good, and creates trans fats, which are very dangerous for you as well. All right, so the second type of lipid is the phospholipid, and phospholipids are involved in creating membranes in cells. Here's our molecule of glycerol, and glycerol is attached to fatty acids. The third spot in a phospholipid is an attachment to a phosphate group, and phosphates are negatively charged. This is something we learned about in the functional groups lecture, the first organic molecules lecture. And the phosphate group is then also attached to something else that's hydrophilic. Now, that gives one end of the molecule a hydrophilic quality and the other end a hydrophobic quality. So this molecule has two poles, hydrophilic, hydrophobic. Phospholipids make up membranes of the cell, both the outer cell membrane and also membranes around compartments in the cell. And they basically separate the extracellular environment from the intracellular environment. The heads flip out, these heads here, toward water, and a bilayer is formed where the heads are hydrophilic facing water and the tails are hydrophobic facing each other. Because remember, anything that's hydrophobic is not wanting in any way to hang out with water. And so they tend to cluster together and are held together by these hydrophobic forces. So this is the exact same picture. And we have, notice, a combination of saturated and unsaturated fatty acids. This combination can be any combination, just like in triglycerides. The more unsaturated fatty acids you have, the more fluid the membrane. And your membrane has to have a particular fluidity. Certain things have to be kept out, and certain things have to be kept in to a cell. And so the combination of fatty acids determines how fluid or how loose this membrane is and its permeability, or what it can allow across the membrane. And we'll look more at that when we look at cell membranes. This is kind of a cool cross-section of the outer membrane of the cell. And this is a layer of phospholipids. And the phospholipids are interrupted by proteins that have different functions in the membrane. Notice I've got chains of oligosaccharides, which are those up to 20 monosaccharides in length chains. And they're responsible for gluing cells together or communicating with cells. But this is the piece that we're looking at right here. These gray, bumpy hydrophilic heads are all over the outside of the cell and also all over the inside of the cell. And then you've got these proteins sort of interspersed within the membrane. Now the last type of lipid are the sterols. This is a picture of cholesterol, completely hydrophobic except for this tiny little hydroxyl group. Cholesterol has a variety of important functions. We need cholesterol. If we don't take it in, cells will make it themselves. We just don't want too much, and we don't want it hanging around in the blood for very long. We want cells to take it in and use it. If high cholesterol means you have lots of cholesterol in your blood. And that tends to aggregate and pile on top of things like saturated fatty acids and cause clogged arteries and veins and heart disease. But cholesterol is important to our cells. And one of the places where it's important is it plugs up gaps in pieces of the membrane that are too fluid. If too much stuff that shouldn't can pass through the membrane, the cell will plug up these holes with cholesterol. Another reason for cholesterol is that steroid hormones are built from cholesterol. So here's the picture of cholesterol, and over here I have a picture of estrogen and testosterone. These are these structures that are initially based on 4 fused rings, and these final molecules have been built from cholesterol as a precursor. There's a whole group of steroid hormones, lipid hormones, including cortisol, which is a stress hormone. And these are different than the peptide hormones because peptide hormones are protein hormones. These are lipids, primarily hydrophobic molecules. Last lipids I want to talk to you about are these essential dietary substances called vitamins, and we take these in via our diet. The fat-soluble vitamins are those that are considered to be lipids, A, D, E, and K. These are primarily hydrophobic and are also classified as lipids, although we don't make them ourselves. The reason why they're important, there's a molecule or a few molecules that are involved in the visual pigments, and vitamin A is a critical component to that pigment. Vitamin E, an antioxidant, which we'll talk more about when we talk about cell division and cancer. This, again, a lipid. All of these points, remember, are places where there are carbons. And wherever you don't see a hydrogen, carbon is actually bound to a hydrogen. D, also a lipid, important in calcium metabolism and bone growth. And K, which is critical to blood clotting, again, a lipid vitamin. So, 4 types of lipids that we discussed: triglycerides, phospholipids, sterols, and lipid-soluble vitamins. Lipid-soluble meaning that they only dissolve, by the way, in lipid. So you couldn't dissolve these in water because they're extremely hydrophobic. That's why we call them lipid-soluble vitamins versus water-soluble vitamins. All right, that is then the end of our macromolecule series of lectures.